everybody, and happy Tuesday. Today we're going to begin our next unit, Chapter 21, Viruses and Prokaryotes. There's actually a couple other topics within the chapter, but it's these two that we're going to be focusing on. And it's actually pretty timely here because it's because of these viruses or certain kinds of viruses that all of us are sitting at home right now. So um, talking about viruses, if this was in class, I'd have you write down 5 things you know about viruses, and if you want to hit pause on your screen, you can do that at this time. And which of these do you know for certain? I'm guessing that some of you said viruses can get us sick. Viruses can infect our cells. Um, and today we're going to start with a little bit of history. How did we first learn about these viruses? Page 574 of the Orange Book, I'm going to read to you a little story here that we would ordinarily do in class. It says, imagine you have been presented with a great puzzle. Farmers have begun to lose their valuable tobacco crop to a plant disease that first appears as a yellowing of the leaves. Eventually, the leaves wither and fall, killing the plant. To determine what is causing the disease, you take leaves from a diseased plant and crush them to produce a liquid extract. You place a few drops of the liquid on the leaves of a healthy plant. A few days later, the leaves turn yellow where you put the drops. You use a light microscope to look for a germ that might cause a disease, but you can see none. In fact, even when the tiniest of cells are filtered out of the liquid still causes the disease. You hypothesize that the liquid must contain certain disease-causing agents so small they are not visible under the microscope and can pass right through the filter. Although you cannot see the disease-causing particles, you're sure they are there. What would you do next, and how would you deal with the invisible? Okay, this is exactly the scenario that led to the discovery of viruses. And this happened in 1892. A person named Dmitry Ivanovsky showed that this certain so-called disease called the tobacco mosaic virus was caused by liquid extract. They didn't know what was in the extract. 1897, another person, Martinez Beijerinck, suggests there's tiny particles in there that are doing it. He called it a virus. By 1935, as chemistry got better, Wendell Stanley isolated the crystals. Okay, the problem at this point was the crystals were too small to see the structure. We really needed an electron microscope, which was developed and used primarily in the later 1930s and 1940s. So now we have a pretty good idea of the basic structure of the DNA of the virus. It's very simple. There's some kind of genetic material, it's either DNA or RNA, surrounded by a protein coat called a capsid. So here are a couple types of viruses. Here is the DNA, this one happens to have DNA in it, and then it has a capsid around the outside. And this other one, same thing, DNA. And then a capsid. There are no organelles, there's no cytoplasm. These things are not cells at all. They don't have a nucleus, they don't have ribosomes, and so on. Very, very simple. 2 basic parts, genetic material and a protein capsid. Some of them also have a cell membrane-like thing around the capsid. Okay? For example, the coronavirus that we're all familiar with nowadays has that, and we'll talk more about that one tomorrow. They're very small, about 10 to 300 nanometers. To put that in comparison, bacteria about 1 to 5 micrometers. So we need an electron microscope to see them. Even the most powerful light microscope will not even show even any structure of a virus. Okay, so genetic material, protein capsid. The capsid is very important because the proteins are what bind it to the host cell. Down below here is the host cell, and they are very, very specific. Okay, if this is an animal virus, it will only attack certain kinds of animal cells because these so-called receptors, the receptors on the host cell only attach to very, very specific capsid proteins. So that's the job of the capsid protein, is to find the host cell. Viruses are completely dependent on their host cell for all processes. Most biologists consider them for that reason not to be alive. They can't function on their own. They need a host cell. They're lifelike. Because they contain genetic material, either DNA or RNA. They can evolve over time. And they can reproduce, not on their own, but they can cause their host cell to reproduce for them. And this is what we're going to focus on today, is how they go through their reproductive, I don't want to say life cycle, we'll just say cycle, okay? Viruses come in a couple different shapes. One type has almost like this little Mars lander sort of shape with the legs and the tail. This is called a bacteriophage. It attacks only bacteria. Here's an actual image of it below. The tobacco mosaic virus we talked about before is this cylinder shaped. And then here's the influenza, and also coronavirus looks like this. This is a sphere shaped, okay, and this one happens to be an RNA virus, okay. Viral infections are very similar to a computer virus in that they come in looking innocent. The computer welcomes them, and then they come in and cause some harm. Most viruses, as we know, can wreck or destroy our computers. Biological viruses are very similar. They come in looking innocent. The host cell here in purple oftentimes welcomes it in because it thinks it's something harmless or something even helpful in some cases. And then the virus takes over the host cell, and it oftentimes wrecks it or kills it. Okay, bacterial viral infections are what I want to talk about today. There's two major types, and we have these little cycle things almost looking like life cycles. There's one type called the lysogenic infection and one called the lytic infection. We're going to start with the lytic infection here. Lytic means to burst open or to lyse. And what happens is in a lytic infection, the virus enters the cell. The DNA causes the cell to make copies of the virus using the cell's machinery. Again, the virus can't do anything on its own. All it can do is give instructions to the host cell. And then the host cell bursts open, releasing hundreds of new viruses. So it looks something like this. We'll start over here. Here's our virus. Here's our host cell. The virus lands on the host cell, injects its DNA, shown in pink here. That DNA then gets transcribed by the host cell. Remember, transcription is when DNA is used to make RNA. That gives it directions then to make proteins and nucleic acids. Here we're continuing down here. And these are all viral proteins and nucleic acids. It then gives the host cell instructions to build new viruses. And lastly, the viruses erupt or burst out of the cell, killing the host cell, and the whole thing can start all over again. This is oftentimes thousands of viruses that come out of a host cell. Okay. The lysogenic infection is another phase, little different though. This time, again, the virus enters the cell, starts out the same, but now instead of killing the host right away, it'll integrate its DNA, which we now call a prophage, with the host cell DNA, and it basically becomes part of it, and the host cell can replicate it, treat it like its own, and copy it over and over again. Eventually, the lytic cycle may occur, but sometimes it may take a long time for that to occur. So that's the green section here on top. Okay, so here again the virus puts its DNA here in pink, but now the pink becomes part of the host cell chromosome. So here we see this big long chromosome with the viral DNA part of it. And the host cell can copy this and make new cells over and over. This doesn't really show it well here, but this can kind of go through a little cycle of its own. Again, the viral DNA isn't being used at all. It's just being copied. And then later on, that host cell can then enter the lytic cycle where the host cell becomes destroyed again. We don't always know what triggers this in these bacterial viruses. Okay, so that's called lysogenic. Lysogenic, again, means that it does not kill the host cell right away. It just becomes part of it and sort of hides in it. Okay, some viruses are what we call RNA viruses. Actually, many viruses are RNA viruses, and that means instead of putting DNA into the host, they'll put RNA in. They'll skip this step. So there's two major methods here. One of them is where the host cell ribosome translates the viral RNA into proteins and new viruses are assembled. And this is an example of, well, COVID-19 right now and the common cold. They're very similar in that respect. Another way an RNA virus might reproduce is by the process we call retroviruses. Retro, as you might know, means to go backwards or to go back in time. And here what we're talking about is the viral RNA is actually made into DNA through something we call reverse transcription. Remember, transcription is going from DNA to make RNA. Well, in some cases, we can use RNA to go backwards and make DNA, and this is what retroviruses do. An example of this would be the HIV virus that causes the AIDS disease. This can then take over the cell through like a lysogenic infection. So an example here would be the common cold, and actually COVID-19 is exactly like this. So what happens is the cold virus will penetrate the host cell, put its RNA in, and this RNA, shown as these little red squiggly single strands, will then cause the host cell to make its own viral parts. So more viral RNA, more viral proteins for the capsids. They get assembled and then they sort of bud off and leave the host cell, which can then go in and affect other host cells. Okay. Certain examples of viral diseases include influenza, or the flu, as we call it, the common cold, West Nile virus, which some of you may know is spread by mosquitoes, the herpes viruses, and then, of course, our common, the one we are all talking about these days, the COVID-19 is a RNA virus. Okay. How do we treat viruses? Well, as many of you know, we have to use vaccines. A vaccine is an inactivated form of the virus which stimulates the body's immune system to fight the specific virus. So what we do oftentimes is give something, part of the virus, usually the protein coat, to a person. They'll get it through an injection or through a nasal spray or something like that, and their body will learn to recognize that virus. And then once the real virus comes along, they can quickly fight it off because they've already got the immune system that can recognize it. We don't yet have one of these for COVID-19. Many people are talking about developing one as soon as possible. There are some other treatments that can oftentimes be used to help with the symptoms of the virus, but one thing we cannot do is treat them with antibiotics. Antibiotics, as we're going to see later on, work only against bacteria, not against viruses. Okay, I'm going to put this video in as a separate link. I was going to show it to you here, but just to keep this video a little shorter, it shows how a flu virus attacks. So again, I'll put a separate link in for this, probably right underneath this video that you can watch. Anyway, we'll stop there for today. Have a good rest of the day.